Good morning. Okay, welcome back. This is episode 2 of the Country Boy Academic. And this morning I just want to say thank you. I put the first video up and I had 33 views in 9 hours, which I thought that was pretty good. I got a little scared because that was kind of a controversial, risky statement for me to make, so I actually took it down and then edited out some names, but the story is still there. And really my point was just to get it out there, that way I feel a little better about my job, and I want to know, and I need the story to be out there, basically. I've told my family what's going on, but you never really know what could happen. I just worry about things getting manipulated before it's too late. And I'm not really a famous scientist. I've been somewhat productive, but it's a slow-moving process in science, and building a career is slow, and you need a lot of people on your side. So I just wanted to say that. Otherwise, this morning I was going to tell you guys a story of something that happened very recently, and it has to do with science, and I can also tell you a little bit about my science in prepping for the story and the way that it unfolded. So I'll just kind of tell you a little bit about my background in science. As I told you in the first episode, I did a biology degree at the University of North Texas in Denton, Texas. And I didn't do any sort of undergraduate thesis or any special projects. I just really liked cell biology and molecular biology. And I found Dr. Benjamin to be a great help and inspiration to me. And I basically decided that I wanted, or at least saw the occupation that I thought would make me happy. And so I had a goal. Plus, as I said, my opportunities or paths forward were limited by other extenuating circumstances. So I just went for it. And so I went to San Antonio. I did a master's in virology. And that really taught me basically how to do science at the bench, how to set up experiments, how to set up controls in a practical manner so that you could get data that were quantifiable and they actually meant something. And so that was 2 years, and it was really good. And Dr. Hans Heidner, he's not a super well funded investigator, but he does really good science, and he's very, very honest person. And I think there, unfortunately, is a inverse relationship between funding and honesty. Oftentimes, not always, but often. So I did my degree, master's degree in virology, and then I started to get a PhD there in San Antonio. I was going to study fungi with Gary Cole. He had a big grant to do that at UTSA, and I started the first semester, and I was taking the core courses and going through qualifying exams. But at the same time, I had reconnected with my ex-girlfriend from Dallas. So I ended up thinking that I wanted to be with her, so I moved back, and I guess she dumped me within 3 weeks. But I started my school at UNT Health Science Center with Anuja Gorpade. I met her, I was talking to an advisor about which lab I might join, and he suggested Anuja Gorpade because she studied HIV in the brain, and I had a master's degree in virology. So we met, and it sort of went naturally. There was a little bit of a story before that, but we won't cover that here. A little bit of drama occurred before I could actually enter her lab at UNT Health Science Center. So I go through her lab, and she studies HIV in the brain, which basically set me up for the postdoc at UCSD. And when I get to UCSD, very early on, Eliza Meslia suggested I look at mitochondria function in the brain, and specifically this process by which mitochondria come together in a process called fusion to make larger mitochondria, or large mitochondria get pinched apart so that they can either be destroyed or they can just be sent to different parts of the cell where they are needed, where energy is needed. And I guess just to recap so that we can bring everybody into the fold, mitochondria are these organelles or structures, like little factories within your cells, that their most famous function is producing energy. They do a lot more than that. They regulate all sorts of molecules that maintain homeostasis and proper function of the brain. So in almost any neurodegenerative disease, you see problems with mitochondria, probably all. I just don't want to say that because I've not looked at all neurodegenerative diseases. But they're very central to cellular function and definitely neuronal function, which makes them very important for the brain. So we started looking at these, at the expression levels of genes that regulate fusion and fission. And what we found was that in HIV-infected people that had neurocognitive problems like forgetfulness, and they couldn't balance their checkbook, they couldn't remember where they're going, these people that we had diagnosed when they were alive, we looked at their brains after they were dead and compared to HIV-infected people that had no neurocognitive impairment. The folks with neurocognitive impairment had less expression of a gene that caused the fission. And so we looked in these brains and we found huge mitochondria, and they were inside damaged neurons. We didn't really look at the other cell types of the brain, but that's what I want to do with my lab now. That was published in 2015, I believe, in the Neurobiology of Disease. Well, since then, as sequencing has become cheaper, other labs have found that this process is involved in multiple diseases. And about a month ago, I received an email, probably 2 months ago now. I received an email from a man in Germany, and the subject line referenced this particular gene, DNM1L, and a mutation in this gene that has been discovered recently, and it's found to cause this gene not to function properly. And the people that have this mutation have very bad outcomes, and it's usually manifest very early in life. So they'll develop normally oftentimes, but then at some point they go into a coma or they start having seizures, and they don't know how to fix it. Many times, I mean, there's only a few cases that have been documented, but most of the kids do very poorly. Okay, so I get an email, and I'm not a clinician at all. I'm a basic scientist. I just happen to publish this paper, and I only work with post-mortem tissues, just to be clear, and then we complement that with cellular models and animal models. Sorry, PETA. So I get this email from a man in Germany, and he says that his son has been in a coma for a year, and they did the sequencing, and the subject line, it has this mutation, and just says basically that the doctors in Germany they're not going to do anything, and they have no options. And I told them straight up, there's nothing that can be done, and I'm a basic scientist, and yeah, there's really just nothing that can be done, and I'm very, very sorry. So he emailed back and said, thank you for replying. I emailed a few of my collaborators. They emailed other collaborators. They all basically said the same thing, that there's nothing that can be done. So I got back to him and I told him, yes, I contacted other folks, and they've all said the same thing, that unfortunately there are no cures or even strategies for this disease. However, if you want to investigate the mechanism, there's a possibility to take the cells from your son, whether it be blood or fibroblasts, reverse engineer them to make stem cells and possibly make neurons and other brain cell types using these fancy differentiating processes that have been worked out over the past 10 years to make little neurons that actually have the mutation and all the genetic background, all the other gene sequences. They should be as close to the boy that's in a coma as possible, as close to any model. And then we could see how the mitochondria, at least in a dish, how they are functioning, and if we can make them function better with any therapeutics. So he said, okay, I'll check it out. Well, I already had a trip to Europe planned. See a girl that actually, all my trips end up involving girls for some reason. But she actually decided we should probably not hang out anymore after I bought the ticket. But this worked out. And I told the guy, yeah, I could come to Germany and try to coordinate the process of getting the cells from the boy and find somebody there to make stem cells. And then you guys could send us the stem cells and we could make neurons. And then we could just test and see if we could find any patterns that with the molecular structure or components of mitochondria. And maybe we could try to solve them with some therapeutics that we have and that we're testing already. So I go to Germany. Wow, this story is going to be longer than I thought. I flew to Norway and then I flew to Amsterdam. I stayed in Amsterdam for 2 days, kind of just getting over jet lag. This trip was planned kind of late, but Amsterdam is always fun. Hung out there for a couple days, and I take an overnight train to Munich, which went okay. The father and a friend that speaks English meet me at the train station. They drive me south of Munich to some little private hospital, and you could see the Alps in the distance as we rolled towards the hospital. And we go to the boy's room, and he's sleeping. Obviously, he's been asleep for over a year now, and the mother's in the room. And it's pretty surreal. I don't work with patients at all, as I said before. And it's very, kind of awkward. But I just tell them, look, I don't have any cures for you. And this is just an idea so that we could try to investigate what the mechanism is, and maybe you could help somebody in the future. So I kind of told them my spiel, like just what we could do. The doctors come in, they are noticeably irritated, but we didn't stay too long because the father and his friend are going to drive me across the other side of Munich to Helmholtz because I'd contacted the stem cell core there, and they agreed to collaborate. So after talking to one doctor who was very dismissive but agreed, then the second doctor, who I guess was more of a boss head doctor, and he sat down and listened to my presentation, but he was also noticeably irritated. He said, do you have any cures for now? The boy's asleep now. I said, no. I'm just trying to, suggesting that we could do investigations that could help people in the future. But I believe there's the doctors, maybe they want the boy out, not because, I think it's a small hospital, and they just have no way to treat him. And I know it costs a lot of money to occupy that bed, but obviously the parents don't want to give up. And if any sort of emergency happens at home, it could be pretty bad. So they drive. I get in the car with the father and the friend. They drive us across town, myself, to Helmholtz, and we meet with this head of the lab, the stem cell core, and she's actually very nice, and she completely agrees to move forward with everything, but she suggests that we take fibroblasts instead of blood cells, and this is important because fibroblasts require stitches. It's an actual biopsy where you have to pull skin cells out with a special tool and then make stitches. And that's just more invasive than blood, where you can basically, the boy already has an IV in, so you should be able to just take blood out. But the reason fibroblasts would be better is because neurons in fibroblasts are differentiated from the same originating tissue. Like after fertilization and your two gametes make one cell, a diploid cell, and it starts replicating. As these cells stack up, depending on what position they are in the stack, they form different layers. And these different layers of this original ball turn into different parts of your body, like the brain and the skin versus the bone, muscle, immune system. Well, fibroblasts and neurons, they come from that same originating cell, which we believe means they should have the same basic genetic blueprint. That would be different from, say, a blood cell, which came from a different original tissue, which substantiates the immune system. So it was important to, or at least the lady that makes the stem cells believes it's important to use fibroblasts, but she'll use the blood cells too. Long story short, not really short, but we get it all worked out. Everybody agrees. They take me to the hotel, give the guys a hug. It was, I'd been in a train all night. I slept one night in Munich, and then I walked around Munich for a day before I took a train back to Amsterdam, stayed there for 2 days, and then I was going to Norway, and then I was flying home. So the next day, I feel pretty good because that was my goal, to meet the boy, get the doctors agree to provide the cells, meet the stem cell core, get them to agree to make stem cells. So all that is done. So I'm just walking around Munich, and I'm from Texas. So I don't know if you've been to Munich, but if you have, there are these rivers rolling through the town. I guess it's one river, and then it splits off into different tributaries. Beautiful rivers. And if you've ever been to the hill country of Texas, it looks very similar with these beautiful crystal clear rivers that are spring fed, and they're very cold. And in Texas, there's all these little German towns. And I suppose it's because a lot of the Germans thought of Munich when they arrived to the hill country of Texas. So I'm walking around Munich the next day, and I see these rivers. So I have to jump in and swim, and it was great. Saw the surfing they do there in Munich on one of the fast-flowing rivers. And then I'm walking through the town, and I see a friend that lives 3 blocks from me here in San Diego, and he's there for a wedding. So that's always very interesting. We had no idea we'd be there. So I hang out with them for a few hours, and my train leaves at about 10:52. And we have a little bit, we go to a beer garden, have some ice cream. That was all fun. And then I head to the train station. So I am waiting for the train, just kind of killing time with my friends. My train was to leave at 10:52, but at about 8:30, I said goodbye to my friends because I wanted to walk to the train station, figure out where my platform is, make sure it's leaving on time, and that all worked out. So then I decided, I ought to smoke a little bit of this weed I have in my pocket that I brought from Amsterdam. That'll make the train ride better. No big deal. Apparently in Munich that is a pretty big deal, and I did not get the memo. So I roam to the park, where I guess everybody does, to smoke their doobies. And I roll up a little doobie, a little bitty doobie. I barely had anything. And I begin to smoke it. One guy walks by, looks at me, and I'm like, okay. Then I see 2 guys walking into the park, and it's very dark. I look back, I'm like, all right, they're coming here. So I get up and start walking away from them. No more than 10 seconds, I hear crazy feet rushing at me. I think I'm being jumped. So I turn around, throw down a bag, and I'm just like, obviously fight or flight, and I'm not that good of a runner, plus I had bags, so we were about to fight. They indicate that they are cops or police officers very quickly. And They were actually really nice compared to American police officers. As I said, I've been in trouble, and they are not so nice over here ever. And yeah, so they, do you have marijuana? We are looking for drugs. You are not allowed to be in the park smoking drugs. I'm like, oh man, I'm sorry. I'm really so sorry. And I felt just embarrassed. And the first thing I asked, because I had a train coming, I said, am I going to jail? He said, no. But there is a fine. We have to take you in. This is your first offense. He asked for my passport. As he said, the Americans are not really the problem they have in the parks, but there is a contingent of folks that are in the parks and doing these sorts of things, and they're like gangs, and it's their job to try to get them out of the park at nights. And I walked right into it, and I just want to apologize to Munich once again. I'm really sorry for breaking your laws, but it was one hell of an experience. So we go into the police station, and they want 125 euros. So I ended up giving it to them, and we actually ended up having a good conversation. They asked what I was doing in Munich, and so it all worked out. And then they took me back to the train station and helped me get a later train. So I get back to Amsterdam like at 3 o'clock the next day. We stopped at Cologne, Germany, and man, that cathedral is amazing. I ended up having a layover there because of a train delay. And that cathedral is amazing. It was Sunday morning, so I ended up just sitting in service. Apparently it's the largest cathedral in all of Europe, or Gothic cathedral in all of Europe. So anyway, I finally get back to Amsterdam after many train switches because the rails were too hot that day or something. And I stay there a couple days, and then I make it back home. And about a week after I get here, or maybe a little less than that, I contact the father just to ask how everything is going. And apparently the doctors aren't even talking to anybody. So none of the things that we set up are happening. So I sent an email with all the doctors copied and just asking if we could push things to get these cells generated from the boy's blood, or fibroblasts, and we've had no answer, and that was about a week ago. So this morning I get another email from the father, and he's just sending me papers that have to do with the gene that is mutated. And it's very sad, but it doesn't look like much is going to happen, and not that much could happen anyway. It was really just to investigate the mechanism and maybe help somebody in the future. But it looks like the doctors over there in Germany aren't too interested, and I understand they don't want to give false hope. So I think I'm just going to leave it alone. If any of you guys have better suggestions, you let me know. Otherwise, I am in lab every day, and I'll tell you some more stuff about my lab later. I just thought that was an interesting story of something that you may not expect a scientist to be doing. Hope I didn't break any international laws. Let's say it was hypothetical. All right, you guys have a good day.